Good morning and welcome back, students. We have studied the lesson reproduction. Today we are going to start a new lesson from biology, that is lesson number 10, human health and disease. So students, what is health? It is related to only physical well-being? No. According to WHO, health is defined as the state of complete physical, mental and social well-being and not merely the absence of disease. So you can see one of the major aspects of health is hygiene. It is a science of rules of health. So to be healthy, it is necessary that we have hygienic balanced diet, clean water, personal and community hygiene, physical exercises, knowledge about diseases, proper disposal of waste, and control of vectors. So again we can see these are the factors essential to achieve good health. Factors essential to achieve good health: balanced diet, clean drinking water, personal and community hygiene, regular exercise, knowledge of diseases, control of vectors, and proper disposal of waste. Let us learn in detail about immunity. The resistance exhibited by the host towards injury caused by pathogens and their products is known as immunity. Edward Jenner developed vaccine against smallpox, and then concept immunity is believed to be started by him. Immunology is a branch of science which deals with the study of immune system, immune responses to foreign substances, and their roles in resisting infections by pathogens. Any foreign substance invading body and capable of stimulating an immune response is called an antigen, and the protective chemicals produced by immune cells in response to antigens are called as antibodies. Let us now study types of immunity. As you can see here, there are two types of immunity: innate immunity and acquired immunity. Innate immunity, inherited from parents and present since birth. It is non-specific defense mechanism. It does not have specific memory. Fast and immediate action. It operates by providing barriers against entry of pathogens. Acquired immunity. It is acquired after birth of an individual. Specific type of defense mechanism. It retains immunological memory. Delay observed between antigen exposure and response. It produces primary and secondary responses mediated by B and T lymphocytes. So you can see in the chart immunity and its types. Immunity is of two types. So these are the two types of immunity: adaptive immunity, also called as acquired immunity, and innate immunity. Adaptive immunity is having again two more types, that is natural and artificial. Natural immunity is again divided: natural immunity, passive and active. Artificial immunity, again passive and active. Let us study in detail about innate immunity. It is a natural defense system of body. Various types of barriers which prevent entry of foreign agent into the body are epithelial surface, antimicrobial substances, acute phase proteins, fever, and cellular factors. Epithelial surfaces. Look at the pictures which shows skin, mucous membrane, cough, acidity, tears, semen, etc. as epithelial barriers. Skin. A healthy skin possesses bactericidal activity due to salts in drying sweat. Sebaceous secretion have bacterial and fungicidal properties. Nasal cavity. Nose prevents entry of microorganisms. Some microorganisms are held by mucus and swept back to pharynx and coughed out. Cough. It is a reflex and it is an important defense mechanism of respiratory tract. Mouth. It has saliva which inhibits microorganisms. Stomach. Acidity in the stomach destroys microorganisms. Urine. Urine eliminates bacteria. Semen. Spermine, that is, it is a polyamine taking part in cellular metabolism, and zinc are antibacterial. Eyes. Tears has antibacterial substances. Antimicrobial substances in blood and tissues. It has complement system and interferons. Blood has more than 30 serum proteins. Microbial pathogens activates complement cascade. It is a part of immune system that enhances the ability of antibodies and phagocytic cells to clear microbes. Interferons are soluble proteins which protect from viral infections. Cellular factors in innate immunity. It has phagocytic cells and natural killer cells. Phagocytic cells are macrophages which engulf or eat microorganisms. Natural killer cells are lymphocytes for defense against viral infections and tumors. Fever. Rise in temperature of body destroys pathogens. Fever stimulates production of interferons and helps in recovery from viral infections. Let us now go through acute phase proteins. Infection leads to increase in concentration of certain plasma proteins called phase proteins. They include C-reactive protein. It is a substance produced by the liver due to inflammation. Mannose binding protein, a soluble serum protein. Alpha-1 acid glycoprotein, a single chain plasma protein. Serum amyloid P, it is an acute phase protein for innate immunity. Acute phase proteins enhance host resistance, prevent tissue injury, promote repair of inflammatory lesions. Let us now study about acquired immunity. So students, we have studied in innate immunity about barriers, antimicrobial proteins, cellular factors, fever, and acute plasma proteins. Let us now study about acquired immunity. It is the resistance that an individual acquires during life. Hence it is called as adaptive or specific immunity. It involves the formation of antibodies to neutralize antigens. So you can see these are the features of adaptive immunity: specificity, diversity, discrimination between self and non-self, and memory. Specificity. produce T lymphocyte against antigens or pathogens. Diversity. It can recognize pathogens or foreign molecules. Discrimination between self and non-self. It differentiates between own body cells and foreign molecules. Memory. The immune system retains the memory of first encounter of foreign agent and then gives stronger immune response to second encounter. These are the types of acquired immunity. As you can see in the chart, there are two types of adaptive immunity or acquired immunity, that is natural and artificial. Natural immunity has two more types, that is active immunity and passive immunity. Active immunity. Antigens enter the body naturally. Body induces antibodies and lymphocytes. Passive immunity. Antibodies are transferred from mother to infant through colostrum, that is first sticky milk, as we have studied in reproduction lesson. Artificial immunity is also divided into two types. Active artificial. In this, resistance is induced by vaccines, for example polio, BCG, etc. Passive immunity. Immunity is developed by previously prepared antibodies using serum from humans or animals. For example, antibodies obtained from horse injected to human against rabies. So students, go through these types. Left side upper one is naturally active immunity and downward is artificially active immunity. Right side upper one shows naturally passive immunity and downward is artificially passive immunity. Let us now learn about cells of immune system. The cells in the body involved in working of immune system like lymphocytes and antigen presenting cells. Lymphocytes. Types of WBCs. Many of them stay in the lymph nodes. You can see in the chart two types of lymphocytes. B lymphocytes, which control humoral antibody mediated immune system, and T lymphocyte, which controls cell mediated immune system. Both these lymphocytes respond in different ways. You can refer the points given in the slide. Let us now mechanism of response of T lymphocytes to antigen. These are four types of T lymphocytes: helper T cells, killer T cells, suppressor T cells, and memory T cells. Just look at the pictures. Functions of T lymphocytes. Helper T cells. It proliferate other T cells, stimulate B lymphocytes and macrophages. Killer T cells. They attack and destroy invading microbes, infected body cells and cancer cells. Suppressor T cells. Suppress immune system against attack on own body cells. And memory T cells. Previously sensitized cells retain for long time in future. You can see mechanism of action of B lymphocytes to antigen. These are sensitized directly by both antigens and helper T cells. Plasma cells produce glycoproteins called antibodies, which are circulated through blood and lymph. And the functions of free antibodies are: first one is agglutination of particulate matter, including bacteria and viruses, and then engulfed by phagocytes. Opsonization or coating of bacteria to promote phagocytosis. Phagocytosis means engulfing of microorganisms. Neutralization of toxins released by bacteria, for example, tetanus toxin. In this picture, you can see functions of antibodies. In this, mass of viruses or bacteria is formed. Then, in this, opsonization, that is, that mass is engulfed by pseudopodia of phagocytes. And then, in this, the toxin is neutralized. Antigen presenting cells. Antigen presenting cells. These cells engulf pathogens and deliver a stimulatory signal to activate helper T cells. You can see in this picture how antigen presenting cells kill pathogens and stimulate helper T cells. So students, today we have studied types of immunity and cells of immune system. Read the textbook thoroughly. Note down important points from this video. Thank you.